speaker to be with us. Let me introduce Dr. Vimesh Patel, who is a group leader for nasopharyngeal cancer research. Dr. Patel is from CARIF, Cancer Research Malaysia. Allow me to read a little bit of his biodata. Dr. Vimesh Patel received his PhD from the University of Bristol in 1993, where his research focus was on understanding the impact of dysregulated EGF and TGF-beta signaling in squamous carcinogenesis. With postdoctoral training and as a staff scientist at National Institute of Dental and Craniofacial Research at the National Institutes of Health in Bethesda, USA, his research goals centered around elucidating the molecular changes that contribute to the evolution of head and neck squamous cancers, and to use this knowledge to develop markers of disease progression and novel cancer therapies. In collaboration, Dr. Patel has been instrumental in helping to design and test nano devices for point of care used for the ultra-low detection of relevant biomarkers associated with cancer development and progression for diagnostic and prognostic application. Okay, today Dr. Patel is going to present his work and share with us his expertise on cancer pathogenesis from observation to clinical application. Please.
Okay. Thank you for the kind introduction. It sounded like a bad report, like a criminal. I apologize for keeping you waiting. The taxi driver couldn't understand my English. So, as Carol mentioned, therapeutics, cancer therapeutics, and translational. So the title here, part of it is from observation to clinical application, which is actually very, very important in our discipline, not only mine but yours. I'm sure you are an element. You have an element of clinical application. Any formulation or from your pharmacy research sort of activities, I'm sure you want to push it to the clinic. And it's the same for us, even in my current setting here in Malaysia. What I'm going to do is take you through at least my timeline through NIH, what I learned, and what I think is very important for me here in Malaysia. Not only for me, but for the group I'm working with, and then to share with you as well what, at least from my perspective, should be important. So having said that, the first slide. I actually stole this slide from the web. It belongs to the Department of Energy in the USA. But I think it's very poignant to take on the message that life in itself is very complex. Simple as it might seem or sound, but if you delve into the nucleus of each and every human cell, there's complexity. So the message here is that you have your DNA, the genome, or the genetic sort of blueprint or the Bible. Then from that, you've got so-called genes which give you life. But before those genes can give you life, they need to go through several transitions. Being sort of from DNA, which is sort of static, to something a little bit more labile to give life, which is RNA and specifically proteins. And it's the normal, sort of, proteins are being expressed not all at the same time, but in their very sort of coordinated manner. Their interactions and so forth to give you that process, biochemical process that impacts the DNA, which is your transcriptional activity. And so specifically, it's those molecules. You've got hundreds of thousands of molecules at the protein level, at the RNA level, specifically at the protein level, that if anything goes wrong, then you're going to get a disease, and that includes cancer. So it's the ability to go from a single cell into the nucleus, tease out the information, and start reading it. Now we're there, we've got the capability. And by reading the message to understand what is normal in a normal process and what is wrong in the disease process, and then to start thinking how this can have clinical utility. So it's this message, I think, if I can emphasize, is for you to remember, not perhaps the most important, but the complexity. And then, of course, we need to apply to a disease process. And then the problem statement is this, it's Malaysia, specifically because we're in Malaysia, but globally, the message here is that why in some countries you've got the North American continent, then you've got the European continent, why in these countries, why the cancer incidence is actually going down. But that's for another discussion. But for us in this area of the world, well, you know, the incidence is actually increasing. There are a number of reasons, you know, socioeconomic and so forth, but that's not for today's discussion. But specifically for us to bear in mind is that because the incidence is increasing, we need to be ready here. So by 2020, this figure has somewhat increased. Malaysia, Southeast Asia is going to be faced with so-called a ticking time bomb, where there's going to be a huge increase in the cancer burden, and it's going to have a huge impact on the health system. And then, you know, are we ready? But again, that's from the discussion. So that's the so-called problem statement. So again, cancer, and then so we focused where I'm at the moment, Cancer Research Malaysia, and one of the cancers that we're focusing on is breast, because one of our team leaders, actually she's the boss. She's interested in breast cancer, and there's two people, including myself, focusing on head and neck because of my background back at in the States and here as well. So this is oral squamous, this is your tongue, the soft tissue in your mouth, and then nasopharynx, which is the small piece of skin, dry skin, at the back of your nose, actually inside your nose, but right at the back, a small skin, piece of skin. That's where NPC arises, nasopharyngeal carcinoma arises, and oral. The nasopharynx is quite interesting. So if I can, again, sort of make a problem statement. So if you're young, if you're Malaysian, which I'm not, either of the two, and if you especially are Chinese descendant, you're more likely to develop nasopharyngeal carcinoma. Okay, more likely. Is it 2 times, 3 times? The only statement I'll make, more likely than your average person in Malaysia. So, which is quite striking. And then to focus on the nasopharyngeal carcinoma problem, you've got some of the Arab countries, including Turkey. The NPC, nasopharyngeal carcinoma, it's totally different than those arising in Southeast Asia. Again, it's not for today's discussion. And then Malaysia has some of the highest incidence, much more higher than actually Indonesia here. And if you think, I don't know what the population of Malaysia is, 25, 30 million, is that right? 30 million, and think what Indonesia has. Malaysia has the highest incidence per population. And then here, if you look down this sort of the different sort of categories from top to right, see how many pertain to Malaysia. All right, quite a lot. And if you look at the very top, Bidayu have been reported to have some of the highest incidents of NPC. Again, problem statement. And think in today's sort of society, in our the ability to travel from one part of the world to the other, mass migration and so forth, then you've got to think of the sort of Chinese descendant or Chinese origin from Malaysia or South China, who actually go to other countries, you know, USA, then you look, you know, Los Angeles, Hawaii, and so forth. So of course, they'll be facing, or they are facing, you know, a problem there with the Chinese descendant developing NPC. Anyway, having said that, that's more epidemiology, and that's not the reason I'm here. I'm here to share you with my expertise, okay? So, head and neck cancer, you know, research and challenges. So if we sort of, you know, look at this column here, this part on the left, with any cancer, if you catch it early, you're very likely to survive. So what is the statement I always give? The best cure for cancer is early detection. Best cure for cancer really is prevention. Preventing, you know, individuals developing cancers. So early stage, this is a huge problem. As you probably appreciate, most cancers are insidious. They do not give any symptoms. They give symptoms which may be, you know, they may be shrouded and so forth, like, you know, runny nose or sort of nasal congestion or a sore throat and so forth. Okay, so if we go sort of, you know, from left to right, the middle part here, if it's detected at late stage, like most cancers are, NPC including, most of the cases we get, I'm not a clinician, but we do collect, we do have a biorepository. 80% of our cases are stage 3, stage 4. Very, very few are stage 1 and stage 2. And that tells us majority are actually detected late. And what happens is that, well, okay, the therapy is still the same here, but look here, poor prognosis. Not only that, I'm going to touch upon this, mucositis. So, can anyone tell me what mucositis is? Anyone? So the condition, mucositis, the reason is I want to bring importance to this. Got someone else?
Situs determine when inflammation will.
No, you're right. So mucositis, after the chemo radio, what it does, it basically destroys, it fries your salivary glands. If it destroys or puts them out of action, either your main salivary glands or the parotid, you can't make saliva. And what happens then is that you have dry mouth, and if you have dry mouth for a long period of time, you're going to get, maybe like what you mentioned, inflammation, you're going to get infection, and it's just a poor quality of life. These guys actually suffer. So just think of life without saliva. Anyway, so if we sort of go from left to right again, 50% of those detected are going to get recurrences and they're going to get met at some stage. 50%, so that's a huge problem. And then if you look down here, well, you know, you can sort of say, for me, focusing on this, limited therapies. Surgery, if you have surgery, think what I've mentioned where the nasopharynx is. Imagine to have surgery, like, you know, a surgeon. Well, they would be very reluctant because it's too close to vital structures, including nerves and so forth. They would be very scared. The only time they would do surgery when it's life-threatening. And then the survival as well, like, you know, close to a year or less than a year. So anyway, fast forward this. For us, our aim is to, you know, try to address this. We're in the middle of validating this peptide vaccine for NPC and oral cancer, but that's not for today's discussion, and neither is this. We have a panel of so-called from next-gen sequencing that we need to validate. And with most things in Malaysia, you can only do things when you have money, so we need to keep applying for MOSTI grants. Okay, all right. So I'm going to take you through this. It's specific for head and neck cancer. It's particularly relevant, well, not particularly, it's relevant to any cancer. So think of the progression, normal to very, very sort of late stage metastatic recurrences. So this part here is predominantly insidious. Here, you won't be able to see it macroscopically. If a doctor looks down your, down your throat or up your nose, they won't see this. They won't see this. They'll probably see this because it'll be very irregular and so forth. This is again carcinoma, where you can see it's already started to invade, local invasion. this is actually away from the main tumor and to the other parts of the body, lymph nodes here. So let me sort of go through this because I think it's quite important. So if you bear in mind this picture I've just sort of painted, then think, well, mechanistically, what are the markers involved? What is involved in the process? Is it cancer stem cells? All right, so here, like, you know, to sort of go from fairly normal, but still not normal, to something normal, but not very normal, if that makes sense. And then, of course, once you get this, then you've got progression. And then, right, think of the, like, you know, the mass public, or the public at large. Wouldn't it be just excellent if there was a marker, if there were markers that we were able to identify those people who might be at risk, either here or at this stage when the cancer is curable, 100% cure rate if it's detected. So if this individual is actually predetermined to develop cancer, then you could put this patient aside and then put them on chemo prevention, chemo preventative regime, to stop them from preventing cancer when, you know, then think of the cost, the life quality and so forth. So, of course, to do this, to get these markers, we need, well, you know, these are the stumbling blocks. I'm finding it right now, the 2 years I've been here, to try and get the tissues here. Everything has been monopolized by one center or another center. No one wants to share. You know, it's actually very sort of challenging, especially in Malaysia. This wouldn't happen elsewhere in the States or so forth. Enabling techniques, we're getting there to enable us to find the targets. Okay, then think of, like, you know, again, markers that we may find that might be indicative of, you know, progression. Then we can start thinking about targeted therapies, you know, different formulations of some of the chemo cytotoxic therapies that are toxic if given alone. Think of everything that you guys might be sort of, you know, doing, including myself. New formulations, targeted therapies, and so forth. And of course, how the know-how, the ability, or the instrumentations and so forth to get the information. And then, of course, once we get the markers, once we get these markers, we need to be ready with cell systems. Well, I'll put epithelial stem cells here, which pertains to this, but specifically cellular systems and animal model systems. Very, very important. I'm going to allude to that quite a bit. And then, of course, then we can start thinking of, you know, how do we stratify patients and so forth to, you know, and so forth. And then, of course, here, training, teaching, you know, web-based. This is actually happening. So data mining tools for the next-gen sequencing and so forth. And then, of course, to, you know, teach the students and so forth. Okay, so having said this, pause. Like the second slide I showed, the slide I stole from the DOD, DOE, is that right? The Energy, Department of Energy. I don't know why the Department of Energy was going with that slide, seriously, on the website. So, if we think about that, then some of those proteins, you know, think if those proteins are being expressed aberrantly by one means or another. So here, if we think of actual signaling networks in head and neck cancers, can be any cancers, the important ones are EGFR. So squamous cell carcinomas, quite a lot, over 80 to 90% overexpress the protein to EGFR, the actual EGFR protein. From the 80–90%, quite a high percentage of those overexpressed are overactive. So you get overactivation of the signaling pathway. It's not normal activity, it's overactivity. Anything that is overactive, it's a disease process. It's not normal. Then you've got a signaling cascade. Each step here is an amplification to take the signal from outside the cell through a cascade, amplifying it until it gets to the nucleus where it has its transcriptional activity. Normal, but in this case, if this is overactive, you're going to get overactive transcriptional activity. Then you're going to get some of these sort of, normal oncogenes having normal function. They're going to become overexpressed and they're going to have oncogenic function, which basically primarily means abnormal. And then, of course, you've got some of the other players, and then you've got signals going to one, in one direction or the other direction. And then I'm going to allude to this pathway quite a bit, all right? But then I just want to sort of let you know, you've got the EGFR straight to the nucleus through a cascade. Then you've got EGFR through the RAS oncogene, the small g, GTPase, and then RAS. If it's mutated, then it's going to give overactivation. So it's going to be overactive and be a driver. In some cancers, a point mutation here is not going to inactivate it, it's going to activate it and become a driving mutation. It's going to drive the cancer. And in some countries, the etiological reasoning behind that, this is mutated and so forth, and it drives the cancer. And also, then, if this is activated, it's going to go through the PI3 kinase in a molecule, then that's going to activate the AKT, important molecule, because it has a huge amount of downstream substrate. And then, if I so if I pause here, go back to the title, observation, from observations to clinical translation. So we were very interested not only in this, because this was very, very sort of, how do you say, exhaustively studied when I was doing my PhD and my postdoc. So we wanted to, because we were at the NIH, we wanted, well, not to do what the others were doing. Then around that time, this was like, if I sort of say when, I'll sound like a very old person. I'll say last century. So when I started my postdoc, which is true last century, this became very, very important molecule. The function wasn't quite known, but they knew that this was important in survival and apoptosis. So we became interested in this molecule with collaborations as well. But if I sort of talk about last century, many of the tools weren't known. The tools were not available, they weren't known, they weren't available, so let's say they were not available. So we were trying to analyze this molecule using old-fashioned radioactivity. So in collaboration with a group in Spain who came to our lab, they were interested in a functional role of this molecule. So basically what they did was develop this mouse skin carcinogenesis model. It's a very, very simple model, but very powerful. And it's localized to the skin. Again, simple but powerful. You need to try it, not try it, but you need to sort of, be involved with it to realize the utility of it. It takes a fairly long time, but once you have, so if you look 30 weeks or so forth, sounds like, very tedious, but if it's ongoing, you can plan your experiments well in advance. So basically what you do, you start with an initiator, TPA. I've even forgotten what TPA stands for, so it's an initiator. It's an ester, I think, right? Anyway, TPA. You just need one application to the back of the skin, and that's enough to start that first hit. But that first hit is not sufficient to start the cancer process. Then you need to give daily dose. So application is DMBA, so it's a carcinogen. So you just paint it. It's in ester, it's in methanol, sorry. Or ether, sorry. No, it can't be ether, so methanol. So all you do, you apply it to the back of the skin and it'll just evaporate every day until you've determined when to end. But specifically, what happens is that you get the early lesions. Go back to that, the scheme I showed, where you've got normal that progresses to carcinoma. This is an early event in mouse, papillomas. Majority of the papillomas actually progress to overt carcinomas, skin carcinomas. So using this model, taking the skin, extracting the protein, and then somehow purifying the AKT. So in those days there was capability by using an antibody that was going to actually pull down the AKT molecule. All we wanted to know the functional consequence, whether it was beta kinase here, we wanted to know was it phosphorylated, was it activated? At that time, we didn't know the activation sites. We knew that if AKT was activated by phosphorylation, then we knew there was activity. We wanted to know the functional outcome. So what we did was actually did an old-fashioned kinase assay involving an artificial substrate to this enzyme, phosphorylating this, incorporating the P32, and then when we see incorporation of P32, then we know that there's the enzymatic activity is active and then activated. So here you can see, so this is the H2B for AKT, and it shows progression. Early papillomas, advanced papillomas to carcinomas, majority of the carcinomas, activated. And then we did the MAP kinase pathway as well to show just parallel. And the group actually published a nice paper in Oncogene, and if you look at the date, this century, 2002. All right. Okay. So what? So we were collaborating with this group in Jesus in Madrid. So from these observations, we said, what about human? This is mouse. What about human? So just as we were going to embark, if you think 2002, antibodies were not available, the phosphorylation sites were identified, then companies, cell signaling, of course, they were very keen to help cancer researchers like us, started developing antibodies specifically to identify the respective phosphorylation sites. Different phosphorylation sites give you different physiopathological understanding. So the antibodies became available. So what we did, the first thing we did, okay, let's get some human clinical samples. These are paraffin archive tissues. Get them and let's stain them. So we did staining. I mentioned the early lesions in one of my earliest slides. And then these are overt carcinomas, but different stages of differentiation. All we want to know the status of AKT, phospho-AKT. And if you can see just very quickly, just looking, normal, very little activated, saying that there's normal activity, highly regulated. Then as it goes into early cancer, you start seeing activated form of AKT. Then of course, when you go into the overt carcinomas, just look at the landscape. These are actually in the tumor islands and so forth. So what? What did that tell us? That told us that AKT activation was an early event. It was an early event. And think of what I mentioned in the prior slide, prevention. If you can find something to either block this from progressing or to stop this from developing, the anti-cancer prevention, chemo prevention. So anyway, so we reported these observations, and then of course, so what? Right? So what? So these are tissue sections. We wanted to work. We wanted some model systems to start doing xenografts and so forth. So in our laboratory, then we had a panel of head and neck cancer cell lines. And basically we just assessed for AKT, like I showed you in the previous slide, but instead of the kinase using radioactivity, we used the available antibodies. So we weren't able to distinguish between the two phosphorylation sites. It was just pan AKT, so activated AKT. So what this panel tells us, or should tell us, is that from this panel of cell lines, there's normal AKT. It doesn't tell you the activity, it just tells you AKT is there. But this tells you in which of the cell lines there's activity, highly activated. So constitutively activated or active AKT. We weren't questioning we weren't questioning then what was leading to the activation of some of these. We just wanted a line to use as a model system. So here H in 6, 12 and 13, and then 26 became our favorite. Me, specifically, work I was involved, this one, because when you put H in 12 into nude mice, it makes xenografts. Having said that, what that meant, we were able to test anti-AKT inhibitors in in vivo setting for developing anti-cancer therapies. This was 2004. Okay. Then we had to think very, very seriously about taking some of the observations into a clinical evaluation. If you think where AKT is, it's very upstream, close to the membrane. But then if you need to sort of look downstream, there are a whole bunch, when I say bunch, hundreds and hundreds of substrates, including a key player, which I'll mention. And so with this knowledge, and I'll bring this to your attention in a moment, we were thinking very seriously about new therapeutic targets for head and neck cancer, and then specifically with rapamycin. All right, so can I ask another question? Does anyone know what rapamycin is? Can someone? Quick. I mean, it's antibiotic. So any other? Yeah, it's an mTOR inhibitor. You're right. Yeah, it's mTOR. But anyway, so you're right. So it's an antibiotic. All right. So it was isolated as a fungus antibiotic, but it wasn't used. It wasn't approved for antibiotic use. It was actually approved because it showed very strong, so basically it was developed as a immunosuppressive therapy. It was much potent, much more stronger than cyclosporine, less toxic and so forth. So when it was actually approved and first indicated for immunosuppressive therapies, what the clinicians found, those patients on organ transplant, rejection therapies, they found that some of these were not developing malignant lesions. Because one of the long-term consequences of taking long-term immunosuppressive therapies is that you develop skin lesions, malignant lesions, either on your skin or on your organs. Usually not malignant, but they're there, they're cancerous, they develop. So it became a huge problem. But the clinicians noticed that those on rapamycin were not developing this. And so then with the NCI, National Cancer Institute at the NIH, when they got to know of this, they started readdressing rapamycin, whether it could be redeveloped as an anti-cancer therapy. So anyway, I've put here new therapeutic targets, chemopreventative strategies, and rapamycin. Okay, so the next several slides, I'm just going to take you through my journey that I was involved in, which was very frustrating at times because it involved very long-term animal studies, but became very exciting when the results were showing fruition. So one such study, it took a long time, like 30 weeks, was developing this oral chemical carcinogenesis model. Not the one I alluded to, because that was the skin. This one was specifically targeted to the oral cavity. And then it was simple in a way that this chemical was given in the water, and so all we had to do was put it in the water at the right dose, let the animals drink, change it once a week, and so forth, and monitor the oral cavity, which was painstaking, but at the same time, paying attention to detail on results and observations, we were able to actually develop this model and utilize it, and there's still room for more utility. So primarily, it's foreign cure, 4-nitroquinoline oxide. When it hits when it touches the normal oral mucosa, it goes through this and so forth. All this has been published not by us, but we were well aware of this. So utilizing this model, what we did was we validated first our experimental design. So in a group of animals, the strain of the animals that we used was very important. We found that some animals were actually showing resistance to this. They were not developing lesions as quickly. They were developing them actually not as aggressively. So they had some susceptibility to show some resistance, which in itself is an interesting question. But the standard white mice, the C50, the strain. Anyway, I've forgotten the strain. It's been a long time. So we had this information and we had a dose in mind. We had done a series of doses and we found that one dose, 50 microgram per ml of water, was actually sufficient just to limit the tumor formation, the lesion formation in the oral cavity. Any more than 50, then you had lesions developing in the throat, the esophagus, and then of course in the stomach as well, thickening and so forth, which we didn't want. We just wanted in the oral cavity. We had this prior information, we did the experiments, and we started. So our experimental design that we sort of optimized and then we stuck with for several studies is start the frontier treatment here for 16 weeks, each week changing the water, and then from 16 weeks, then instead of carrying on with the carcinogen, just replacing with regular water, and then monitoring these until the end of study. End of study was determined by the animal protocol, and if you look here, the animal just started dying. All right, so just pay attention to this just briefly. This determined when we finished the study. So we had a broad idea when the lesions were about to develop, even here. And then the lesions, those early lesions develop into carcinomas, and the animal's actually suffering and actually dying from the disease. And this is actually just the burden, the actual per mouse. So just counting, painstaken, and this graphic is quite dramatic to show that there is increase with progression. Anyway, so what? Again, so there have been many studies, even prior to when we started, there were many studies actually having reported the 4NQO model of oral carcinogenesis. So what? Okay, so again, go back to the last slide that I showed, clinical utility. So we said, okay, rapamycin, mTOR, and so forth, we had prior information. We said, okay, let's use it. So this slide is just to show that, so this is clinical features. Then if you take these slides, the tissues, and you can actually look at the histological slides. So the carcinoma broadly followed what we see in the humans and so forth. So we said, hey, actually, the progression here, when you take the animals off, the carcinogen, they progress like they do in humans. So we said we had a mouse model of oral, oral cancer using chemical carcinogen, mimicking cigarette smoking. So we tried to massage it to our specific objectives at the NIH. Okay, so back, I'll repeat myself somewhat, clinical utility. So then we decided, well, okay, what about using rapamycin to prevent progression from 16 weeks to 22 weeks, right? Would it prevent progression? Go back to one of the earlier slides, looking for AKT in early lesions in human clinical samples. The earliest sort of lesion showing activated form, so we thought, let's check in this mouse model. So all of this is just decoration. You just can't, stain for just one marker and say that. You need to make a story. So we chose some of the obvious. The ones that we chose were, you know, COX-2 was inflammation, P53, well, the antibody was working well, BRDU to show proliferation, and then so forth. So these two are tied very closely. Phospho S6 is downstream of AKT, activated AKT. If this is activated, then AKT will surely be activated. If you just focus here, it tells you that if you look at the non-neoplastic and dysplastic early lesions, highly activated. So then that told us, perfect. Okay, let's go and test if rapamycin can inhibit this and prevent progression. So what we did with the model system, followed the same strategy up to 16 weeks, took the animals off for NQO, whatever the number of animals, we divided them into two groups. One arm receiving rapamycin every day for until the end, and then the other arm vehicle control every day. And this is what we found. So end was determined by the control group actually just dying off, and then so forth. So before everyone died off, we sort of decided to end everything and retrieve, after euthanization, retrieve the tongues here, and then actually start measuring. Not measuring, counting, so painstakingly measuring. So we had a pathologist who helped us do this. So multiplicity of squamous cell carcinoma is actually counting, like how many per tongue, and this tells you that's much higher in the control than rapamycin. Then this is the tumor area. Basically, if this would be one tumor area, and you know this is the sum of all of these, and then you can do what you know, sort of mapping and so forth, just to sort of get that figure panel to give you that impact to try and get your paper published. So what that told us, prevention, right? Identify if you have a person at risk, and for instance, if AKT is activated, then you can think of that person giving him a low dose of rapamycin as a preventative regime, and then monitoring on a regular basis to see that the cancer or the lesions, or you know, those patients are not developing any malignant lesions. Okay, so go back to why I'm giving this talk, and then the title, Observations to Clinical Application. And so with this information, we were exploring doing a clinical trial. By the time I left, it was still unexplored, but the group is still actively following chemoprevention, but not with rapamycin, but another regime or another agent which actually targets the AKT-mTOR pathway. It's not rapamycin, it's metmorphin, which is the anti-diabetic therapy. This is actually raising a lot of interest. So the group in USA is actually following this as a chemopreventative measure. Okay, so what? All right, so mouse model. What about humans? So you can do a clinical trial in mice. All right, what about humans? So I mentioned AKT, I mentioned some of the downstream, mTOR is huge. mTOR is huge. All right, and then rapamycin, well known as a target of mTOR. It binds to an intermediate protein, and that complex then binds. Since then, you've got direct inhibitors of mTOR. mTOR now is very complex. It's not mTOR alone. It's TORC1, TORC2, there's TORC3. All different subsets of mTOR have different downstream functions, which is still relatively, well, not unexplored, but it's raising a lot of interest in metabolism, calorie restriction, and call it what you want. So then if we look at phospho S6, I mentioned the most downstream target, and I'll mention this again, we ended up including this in the clinical trial as a surrogate marker of AKT activity and mTOR activity, the most downstream, because the antibody is very good and it's very sensitive. All right, so then we were exploring rapamycin, if rapamycin could be re-indicated for head and neck cancers. You've got rapamycin, FDA approved, no patent, but then you've got the rapalogs, which are sort of different variations of this, but they all come with baggage. Baggage means they belong to a company and they charge and so forth, but this is very cheap. And there's a clinical formulation of this as well. Okay, so with this information and the information that I've already alluded to, and I mentioned one of the cell lines or several cell lines overexpressing, showing high levels of AKT activity. All right, so what we did, we put these cells in nude mice. Of course, we knew that these mice would then develop lesions because we've transplanted human cancer-inducing cells. And then what we did was, just treated them with rapamycin. And if I can just pause it for you, sort of people, youngsters. So we started working with rapamycin I'm almost embarrassed, 1995 when I first joined. This is when NCI gave us rapamycin saying that we think it could be redeveloped for anti-cancer therapies. In those days, 1995, the only sort of analysis, or if you wanted to do drug evaluation, and this is what I'm noticing here in Malaysia. Well, what is it you do? You get some herb or you get some plant extract, a natural product. It may, anti-cancer therapies, and they say, yeah, it kills cancer cells. So which assay did you do? Oh, we did MTT assay. All right, we were doing the same assay, 1995, which was a wrong assay to do. Wrong assay. And this is what I'm telling people here. Don't restrict yourself to MTT. I read grants said here, and this is what they want to do. MTT, apoptosis. We were doing this 1995. We've got some good papers, by the way. But for rapamycin, what happened? We threw rapamycin onto our cells to do an MTT assay. Nothing happened. Cytostatic. We saw no cell death. Right? So we said we didn't know what to do next, and this is exactly what's happening here in Malaysia. Sorry, maybe it's not happening here, but definitely elsewhere. elsewhere they say, well, so you need to know much more. So what happened was that rapamycin in vitro was cytostatic. Right? Cytostatic. The cells were happy. I mean, not happy. They weren't doing anything. But they weren't, rapamycin wasn't killing, did not have that cytostatic. But since we were new and so forth, so we dropped in 1995. And if you look when we published this, 2005, just imagine if we had done this experiment in 1995. I wonder if I would be here now in Malaysia. Okay, so it was a big wake-up call. We were very reluctant to do this study, me and my colleague here, he's first author, so postdoc. So we said, okay, well, let's just do it. Let's just put some cells into mice and treat. Then we started treating every day, and if you look by 9 days, I said, well, what? And then 19 days, and of course we followed. We repeated this several times, it was real. We repeated it in several cell lines, it was real. So we got a nice paper out of it, by the way. So this, together with the other information, our boss then actually put together a clinical protocol to do a clinical trial. So the clinical trial was a phase 0, a neoadjuvant, and we insisted. So before we went to medical oncologists, they said, well, you can only do this on those oral head and neck cancer patients who stopped responding. But we knew that was the wrong population. We insisted newly diagnosed. And so we managed to convince a cancer center to work with us, and this is indeed what's happened. So a clinical trial, the trial is finished, but it's not closed because they're following up the patients. 20 patients, newly diagnosed, there was inclusion criteria, 20 patients included, so in the inclusion criteria, went through newly diagnosed, imaging, a piece of biopsy, then they went on a daily regime of rapamycin, oral formulation, every day for 3 weeks. At the end of the 3 weeks, imaging, a piece of biopsy, then they went to get their standard of care, because you cannot deny patients their right to standard of care. And the standard of care for that center was surgery, and they had surgical and so forth. So these patients are being followed. So the study has not been reported, but in ASCO and so forth, there is promise. There's promise, at least for squamous cell. So I just want to reiterate, so observation to clinical, this was quite exciting for me, and I just want to share you with this. Okay, all right, so Carol, how much time? Just stop me when we need to know.
So much time. Just go on and on and on. It's okay.
But then these guys maybe have better things to do. Okay, so anyways, I just want to share. So okay, then okay, this was exciting. Then of course you can't sit back. What's next? So we were really hungry, and so if I can share this with you, so what we wanted to do was think about, I mentioned prevention, I mentioned about progression, and I mentioned about sort of, how do you sort of, prevent cancer from progress, sorry, in the last slide, how can you cure cancer? This one, I've mentioned prevention, I want to now address progression. Imagine the clinical question, the patient is newly diagnosed. The question always for the clinician, the surgeon, the imaging, and the tumor board, what the medical oncologist wants to know, has it progressed? Has the tumor gone beyond the boundary? Has it invaded? Has it progressed? Is there progression? Because if not, then that patient probably will get a different treatment, or should get a different treatment, and get a reasonable chance of surviving. So the question for us was, well, can we use our sort of, knowledge, our tools, our expertise, our mouse models, our enthusiasm, our sort of, whatever it is, we had then, like keep the momentum moving. So we said, okay, primary tumor, then if it's not progressed, can we prevent it from progressing? But before we could do that, or before we proceeded, we sort of said, well, in parallel, and this is the other thing I emphasize to people I work with, you can't do one thing at a time here in Malaysia, even though resources and so forth, you always need to be thinking in parallel and so forth. So in parallel to us developing the mouse model, and I'll go to that in the next slide, we said, what about the clinical, the human clinical sort of, side? So we said, okay, no problem. Let's get the primary tumors that we know that they've progressed to the lymph nodes. So we had some primary tumors where we knew that the progression had occurred to the lymph nodes, okay, so-called. So this, just to reiterate, so these are primary tumors, just to show you, H&E. CD31, and I'll allude to that, think about how tumors metastasize. CD31 is blood vessel, markers for blood vessel. LIFE1 is a marker for lymphatic vessels as well. These are conduits for cancer cells to travel and metastasize. And then our favorite molecule, phospho-S6, and we see that it's activated and so forth. Then we go into the lymph nodes from the same patients that we knew had primary tumors, but with progression to the lymph nodes. So again, H&E, cytokeratins to show, well, the usual. And what is the usual? And I've forgotten now. Okay, cytokeratins to show that the tumor cells are in the lymph node, and cytokeratin is specific for epithelial cells. Then phospho-S6, right? It showed us that there's high activation in the actual tumor cells in the lymph node. The lymph nodes run your neck area. If the primary tumor is in your tongue and you've got your lymph nodes where you have tumor cells, then you know you've got progression. And this is just basically measuring this and giving an index. So just by looking, there's high activation of the AKT-mTOR pathway. and then there's high expression levels of these two important markers. Okay, so this gave us figure 1 or figure 2, doesn't matter, but it gave us information that we needed to proceed. And of course the animal model. And so to model the primary tumor cancer progression to the lymph nodes, all right? So then you need to think of putting human oral cancer cells in the tongue, and then you need to monitor how they progress to the lymph nodes. Ideally, in our dream, we wanted to see cells like go from here to the lymph nodes. All right, but anyway, in our dreams, we tried, but then we knew it's just, you know, we did have the instrumentations, but we just didn't have the know-how. Anyway, we used India ink, which is a colloidal ink, just some dilution. The animal was alive under anesthesia. We opened up, we injected, and then we were very quickly, upon injection, we were able to sort of see the trace to the lymph nodes. Here's the lymph node, and so forth, telling us where the drainage basins were, so we could quickly open up and take those lymph nodes out. And taking the lymph nodes out that had ink sort of injected, and you can see this is your very classical subcapsular sinus highlighted by the ink. And then, of course, we injected, we waited for, this is without ink, tumor cells. Then after several weeks, we had that information. We knew when we would get progression of the lymph nodes. Taking the lymph nodes out, then doing H&E gave us some indication that you've got lymph nodes which have human cancer cells. And of course, staining for our favorite markers, like one. Then I think one of the slides has phosphorus S6. Anyway, this was our animal validation. Then so what? I mentioned about injecting the cells. I mentioned about knowing when we have tumor formation and when we have metastatic lesions. What we wanted to know was when we knew that the tumor was very early, after several days of injection, if we put them on a regime of mTOR inhibitors, were we able to prevent progression? All right? So this is just the primary tumor vehicle, huge tumor burden. I think this is a lot easier. And then with the two, rapamycin and one of the analogs, equivalent benefit. And this is just sort of visuals to tell you. And then taking out the tongue and the lymph nodes, we were able to actually stain and put some sort of measurement just by looking at the staining and with the pathologist helping us to give a score. Or if we look at a classical sort of primary tumor in the tongue of a mouse injected with the human cell lines, we can see the primary tumor with our favorite marker is activated, and then the lymph nodes, the invaded lymph nodes, activated. Here's the tumor area. And then if we do our immunohistochemistry, if you look at the tongue of the untreated control, you've got high activation of our favorite markers. And then the equivalent, including the metastatic lymph node. If we include, if we look at the treated rapamycin and the RAD001, you have, I would say, almost zero, but there's a huge reduction of staining, telling us that there's been inactivation of the AKT-mTOR pathway and thus less invaded lymph nodes. And again, the treatment strategy, as soon as the tumors started developing, we put them into, randomly sorted into the 3 different groups and treated each and every day until end of study, 21 days. Okay, that told us, clinically, that told us that rapamycin can be used as a tumor, so as an anti-progressive or anti-progression sort of strategy or regime. tumor, as a patient with primary tumor, not yet progressed, put them on a low regime of rapamycin or mTOR inhibitors, then that patient has a chance to have his primary tumor reduce and preventing metastases. So maybe if I can make a point here, the biggest killer of cancer is metastases. As soon as you get progression and metastases, that will kill you. Okay, so change tracks here. So I've mentioned anti-cancer therapies for prevention, actually preventing, or as an anti-cancer sort of regime, and preventing progression. I mentioned about mucositis in one of the earlier slides. It's a huge problem. We had some inkling after, so we published this, just before I left, or so I don't even know, 2010, 2011. So we had some inkling that if you inhibit mTOR, it may be targeting stem cells as well for sort of inactivation, or preventing stem cells from being activated to have their function. So the stem cell compartment in the oral cavity of the mouse or the human, it's relatively still unclear. It's not that it's not unexplored. There is a huge sort of line of investigation, but we're still not able to identify where the stem cells reside in the tongue. In the skin, it's well known. It's in the sort of, the hair follicle compartment. But we had some inkling. But with the inkling, you need to show. So just get some data. So we were working very closely with another group in the institute who had a mouse model of mucositis. They were working on a different regime, on a different sort of angle. They were using KGF, gene therapy, and so forth. So we went to them, all we said, we want to collaborate. Please collaborate with us in a mouse model, because we don't want to write an animal protocol and so forth. So they agree. And all we said was, with your mouse model, we want to treat. They were using radiation to induce mucositis, and then standard of care for head and neck cancers is radiation therapy. So what we said was, with a sort of experimental design, they agreed and so forth. Then, talking and talking is not sufficient, so let's do it. So we did it. Together with Anna, we actually did this experiment. If you just focus in this animal design, we had two arms, or two design settings. One design was just one dose, and another design was fractionated dose. So the one dose obviously got a higher dose, just single, and here fractionated over 5 days. The caveat being is that in one group, or one arm of the group, just before, an hour before fractionation, radiation, the mice were given rapamycin, the standard dose, IP, each 1 hour before each fractionated dose of radiation. Then here the animals were left for another further 5 days. Then the samples were collected. So basically the tongues were removed. And then they were stained with violet blue, I believe, yeah, violet blue. It's just a stain, and basically what it does is, if the skin or the wet tissue absorbs the stain, then it's lost the barrier function, the normal barrier function. If it doesn't stain, then it's still maintained. Here it was very clear. So these tongues developed mucositis, ulceration, within 10 days, and these were actually clean. And then we put some index and so forth, and then there's some histopathological evidence. And then here, all we sort of said, there's erosion, and someone mentioned erosion here of the wet mucosa, and here it just seems normal, you've got total protection. So what we said with some mechanism, what we sort of said, rapamycin, at least maybe taking those stem cells in the basal layer into a quiescent state, and basically, if you just look at maybe this is high magnification, I believe. No, this is high magnification. This is the upper part of the epithelium. We're looking at red here. There isn't. All gone into quiescent. And if you look at the normal, you've got quite a lot of red color, which is the activated. Put it into quiescent, and then very little, and so forth. But if you look in the radiation treated, activated, and basically it's showing that the stem cells have actually, you know, been either destroyed or having an abnormal function. So we do not know function-wise. All we know that clinically we can use these observations to prevent the onset of mucositis in cancer patients receiving radiotherapy and chemotherapy. So what we're trying to do at Cancer Research Malaysia is use these observations to go around hospitals. Actually, we went to Hospital KL, Hospital KL, to talk to a radiation oncologist and sort of showed this, and they're interested. We sort of said a phase 0 or a neoadjuvant, like patients newly diagnosed, before they get radiotherapy, is there a way we can give them a safe dose of rapamycin over 5 days? Doesn't matter duration of the fractionated dose. So there's some talk. So we are very keen to explore this. Alone we can't do this. So how much time? I can stop here.
Just 1 to 2 minutes. Can I just ask you, how did you administer rapamycin?
Just IP, just standard IP in these lines. All the administration was done very simple IP.
Rapamycin is water soluble?
It's soluble. So you do need to take it through some. The stock solution it's in ethanol, 100%. Then you can take the stock solution and put it into your working solution in whatever, do PBS, DMEM. It's actually, it doesn't come out of solid solution. And I was going to take you through some of my intravital. We did a lot of drug delivery formulation. We used carbon nanotubes in those days, and then we labeled them with cisplatin, not labeled, we decorated the carbon nanotube with cisplatin as well as imaging labels, and actually in real life, growing the tumors, administrating, and we found intravital in real time. The nano formulation was able to actually come out of the blood system around the tumor, and did it have a killing effect? It had some effect, but we just showed this anyway. We got a nice paper out of this. And then this is very, to study tumor angiogenesis. Again, mouse models, it's very, very possible to study very nicely in a very simple sort of mouse model. This is a nude mouse, and taking the skin here, you actually gouge out windows. You take the upper part of the skin and put in glass cover slips that you can monitor, in this case was tumor angiogenesis, but you can monitor anything you want, but you need to have that question. For us it was sort of tumor cells and then anti-angiogenic therapies here. And then this is what it looks like in the mouse. It's all under anesthesia. They're comfortable, but they recover quite nicely. But you need to monitor, so you can't, you need to monitor. But of course, you need to have the right animal ethics and so forth, So our question was, our experiment design was tumor cells. And I'll go through this. So tumor cells. So we used colorectal. This is actual real experiment. Colorectal cancer cells, which were labeled with tomato. We put them into this skin chamber thing I've quickly shown. And if anyone is interested, I can spend more time later on and so forth. And this is over 1, 3, 8, and 12. We're able to monitor this in real time using intravital. And this is with a experimental anti-angiogenic inhibitor. You can see from day 1 to day 15, there's a huge reduction. And if you can show something like this is very compelling, rather than in vitro using standard anti-angiogenic sort of in vitro properties that I'm still coming across here, which is very descriptive. It doesn't actually give you like is it real or not, and then and so forth. Just very quickly, and then very quickly, us in Malaysia, where we are. So we have a fish here. Fish actually have blood vessels. They develop nice blood vessels, well studied, and they are also lymphatic vessels as well. Think of what I mentioned, the lymphatic vessels, blood vessels, these are conduits for tumor cells to travel. And so with these two fish, which we have, by the way, Cancer Research Malaysia, and it's open for collaboration, by the way. And then we can start thinking of anti-angiogenic compounds as well as anti-lymphangiogenic compounds as well. All right, so we have this. And then, of course, we have Malaysia. I have no idea why. Can anyone explain why Malaysia is called this?
Sunderland.
That's what I said. No one's actually given me Anyway, so but it's not for me to. So it's mega biodiverse. So basically, it means natural products, and so using our access to natural products and compounds from natural products, we've started exploring anti-angiogenic and anti-lymphangiogenic. And just to quickly show you, so our limitation in Cancer Research Malaysia is our imaging capability. This is using an inverted, which is still, I would consider, not optimal. You can't get a front cover to a journal like Nature or whatever with images like this. We need, and so this is something strongly advocating, to our boss here. And then, of course, modeling human cancers. So we've put in human cancers into the fish. This is doable. People have done this. It's optimized. We want to optimize it in our laboratory. So you label the cells. It can be any cells, human cancer cells. Label them that you can monitor against the red fish or the green fish. And here, I don't know if this is real or not. We do not know, but we say, is this normal blood vessel development, or is it something due to this? And so it's something that we would like to explore further. And then this is actually Sean that you met when you came. He actually developed this double transgenic, which we also have. So then, we have identified a few. I'll just give you an example of this. This is the lymphatic sort of vessel here. So this is a live one. I mentioned live one several slides back. It's a marker of lymphatic vessels. So they've just used the promoter to live one to actually label the lymphatic vessels. And then with the treatment of this here at 20 micromolar, we're noticing that there's a defective development. And so we're interested in chasing this on how specific this is or not. It's yet to be tested. And then we're looking at some other readouts like defective development, indicative of the targeted pathways, and so if I can emphasize this, whatever you might be doing, you need to have it translated into a relevant model, and as many as possible. And then this is very important as well. And then anyway, okay, that's it. Sorry.
Okay, thank you so much, Dr. Patel, for very informative lecture just now. So maybe we can open for question and answer.
Taxi driver. So For me, if I have to say it, I might be maybe not saying very nice. This is confocal. This was done in New Zealand by the postdoc. No. So this is totally. So we want to attain to this level. So what we advocating to our sponsors, our funding agencies, this is what we need. It's not going to happen to MOSTI. MOSTI are not interested. So we are reaching out to Welcome International and so forth, saying that we need a confocal. We don't need any confocal. We need a confocal that will suit us. This is confocal, but if you compare this with this, so and then if you compare this, you can't publish, right? So I'm telling my boss, I said, this is our bottleneck. If you want Cancer Research Malaysia to be the best in Malaysia, we need to do something. So we can only go as scientists to try and do the best we can, but we need to also say we can do better if we have better access.
Injection of the oral cells etc.
Yeah.
The oral cancer cells is human.
Yeah, they're human. So
Is there no rejection?
no. So so if I can say so, I'm not a fish expert, but yeah. No, so these are the things you learn. So it's excellent when you have the capability of learning. So Sean, not learn. So having the ability to learn. So at this stage they are athymic. Their immune system isn't developed. Their T cells and so forth is not developed. So they can actually. As soon as they get beyond 6 days, I believe, or maybe I don't know, but it's around about 6 days, then you start getting issues. But you can start doing things like this in adult fish because they have the mutant form of the RAG1, RAG2, which is again, so the nude mice are RAG2 negative, and so they've developed a fish equivalent of a nude mouse. So then you can start putting in human cells into adult fish. So this is very cool.
You mentioned about translational research on lab and bench, clinical trial. From your experience in just asking, after people have done all the tests, and going to animal studies with mice or whatever, is it necessary to work on primates before it goes to human trial?
To be honest, I'm probably not the best person to answer this, but from my experience, no, preclinical observation, as long as these are very simple mammalian model systems that show whatever effect that you want to show, then this is sufficient. Right? As soon as you start getting primates and so forth, then for me, primates and so forth is for safety. Right? Safety, like is going to be safe, tolerated, so forth. And then if you think, what is the NIH trying to do now? And definitely no more primates. They are actually putting all the chimpanzees in the NIH sort of umbrella to retirement. They've actually put them out to retirement homes. There's no primacy involved. In the old days when I started, the HIV vaccines and so forth were, but not here. So we went to Monash, and one of the professors and says, if you need access to monkeys, I have a group in, so we don't need to go. So I'm understanding we're all on the same sort of level here. We want to go to translational research to try and go from bench to bedside in the quickest possible way. So it's those observations, those striking observations in as many model systems as possible. That's sufficient. When I say as many model systems, meaning different human cancer cell lines in the same animal, or if you have, like what I've alluded to, different kinds of squamous model systems like skin, chemical oral, they all give rise to animal, mouse squamous cell carcinoma. that is sufficient also. Then you can start thinking about genetically modified mouse, where markers that you know have been deleted or activated to give you that so activated RAS, KRAS for colorectal. There are mouse models. Perfect. Anyway, I mean, you have my contact details if you know, I don't know, if you're too shy to ask.
There are quite a number of us on some cancer research. We are all behind you, next to you.
No, anything. Seriously, so if I can give an advice, because I mentioned I started at NIH in 1995, okay, so if I can share this with you, I go from one environment, which is in Bristol, like, you know, how the British work, or don't work. So I went to the NIH, and I was like, you know, just totally in awe. I thought, where am I? I mean, you know, out of my league here. So one Sunday, I was sitting there thinking, you know, my God, I've got nothing better to do, and I mean, you know, country probably, you know, didn't want to be in. I was still relatively new, and then the boss actually came down the stairs Sunday, which totally caught me by surprise. He said, "Hamish, what are you doing?" So I couldn't answer him. In those days, we didn't have sort of individual computers. I was sharing a computer, and I was looking at the football scores. So he said, "What are you doing?" So I said, anyway. So he said, "Look, this is NIH. You know, it's full of smart people. This is you know, emphasizing it. This is the NIH. You know." So, and I said, "There are smart people. Go and find them. Use them." And he just turned around and went away. So then I thought about it. I said, well, if I don't make the effort, if I stay shy, reserved, and I don't go and talk, like let people know who I am, let people know what is it I want to learn, or what is it I want to sort of, you know, talk to them about. No, you're not going to know. You're not going to find out. You're not going to find. I forced myself, then I realized, you know, there are some very nice people, very smart but very nice people. So the cliche I sort of used when I started getting students you know, and so forth. I said, look, the friends you make in this environment will invariably end up being your long-term friends. Right now, I can say all those friends I made there in Kyoto, Tokyo, Seoul, in Thailand, China, Shanghai, back in Spain, South America, and we stay in touch. Every now and then they email and say, do you have this? Well, I have this idea. Well, can we work together? So we're still able to collaborate. This is excellent. And when I go to those countries to visit, you know, I don't get the chance to go like personal visits, like, because, you know, in China I would get intimidated. So just because of the language. But I need to go to Hong Kong. But anyway, but if there's a meeting or I happen to be in the area, they say, no, come, we can organize like a talk and meet the people, and then, you know, it makes it quite enjoyable. You know, so you can still like see what they're doing. And some of these friends I made a long time ago, they're professors, head of, you know, whatever it is, you know, they're busy, older and so forth. So it's excellent. You know, excellent. So I'll encourage you guys to be less shy.
I think that's all. Thank you very much, Dr. Patel. So just some small token of appreciation from our side.